Okay, here we are, chapter 26, the colonization of land by plants and fungi. For John Tyler Community College, my name is Mr. Sparks, and I will be guiding you through this lecture. If you have any questions, feel free to contact me on my email or by phone if it's an emergency. Okay, here we go. For more than the first 2 billion years of Earth's history, the terrestrial surface was lifeless. It was just barren rocks, perhaps covered by bacteria. Cyanobacteria likely existed on land 1.2 billion years ago. Around 500 million years ago, small plants, fungi, and animals emerged on the land. The animals were likely the arthropods related to marine crabs or organisms like that were evolved for an amphibious life. The first forest formed about 385 million years ago. Okay, the first forest with large trees. The trees were made of tree ferns, like we see here in this tropical forest. You can see off in the right-hand corner of this slide. There's a tree trunk you can see on this fern. This is a classic tree fern. In the ancient forests of the Carboniferous era, the trees were made. They were club mosses, which is a form of primitive plant, and they grew into large tree structures. Those types of trees are extinct nowadays, but here we see traditional, or this is a modern scene, so we have modern angiosperm trees. And we're going to talk about all the evolution of all these plants throughout the course of this lecture. Although not closely related, plants and fungi colonize the lands as partners before animals arrive. Plants supply oxygen and are the ultimate source of most food eaten by land animals. Fungi break down organic material and recycle nutrients. This is an important process, a service that is performed by fungi. Okay, you can see here in this phylogenetic tree, here's a common ancestor, an early protist, which gave rise ultimately to charophyte algae, and then plants, modern-day plants, came out of the same common ancestor, whereas fungi and animals, believe it or not, fungi and animals shared a common ancestor, I guess about 500 million years ago or so. So because we can tell this because of certain protein and molecular characteristics that the fungi and the animals share. So you'd think fungi would be more closely related to plants, but in actuality they are more closely related to animals. Fossils show that plants colonized land more than 470 million years ago. Green algae, called charophytes, are the closest living relatives of land plants. Many characteristics of land plants also appear in some algae. However, land plants share certain distinctive traits with only charophytes, including rings of cellulose synthesizing complexes and structure of flagellated sperm. So these characteristics indicate that there is a close relationship between land plants and charophytes. Now we're going to describe the charophytes in a little more detail. Okay, here is a circular set of proteins that are embedded in the plasma membrane that are found only in land plants and charophyte algae. So charophyte algae and land plants, this is one of the proteins that indicates that they share a common ancestor. Comparisons of both nuclear and chloroplast genes point to charophytes as the closest living relatives of land plants. Note that land plants are not descended from modern charophytes, but rather share a common ancestor with modern charophytes. This is similar to how humans are not descended from chimpanzees, but rather share a common ancestor with chimpanzees. So this is common ancestry. This is a trait of phylogenetics that we learned about in the earlier in that lab and in some earlier chapters in this book. Chera vulgaris is a pond organism. It's a complicated, structurally complicated green algae. And these little red dots in here are the spores, the next generation of this algae that will be released into the aquatic environment and hopefully find their purchase and grow into new Chera vulgaris. And this is Coleocate orbicularis. It's a disc-shaped charophyte that also lives in ponds. So these are two of the primitive, these are two living examples of the type of algae that shared a common ancestor with what are now modern-day plants, land plants. Here's the Chera vulgaris, Coleocate. Adaptations enabling the move to land. Okay, certain things had to, adaptations had to be readied in order to allow plants to inhabit the land. In charophytes, a layer of durable polymer called sporopollenin prevents exposed zygotes from drying out. So the spores and the zygotes in the charophytes are covered with a substance, a protein that inhibits moisture loss and prevents them from the zygotes from drying out. Sporopollenin is also found in terrestrial plant spores. The movement onto land by charophyte ancestors provided unfiltered sunlight, more plentiful carbon dioxide, and nutrient-rich soil. Land presented challenges such as a scarcity of water and a lack of structural support. So there were lots of opportunities to be had. There were lots of vacant ecological niches on land, but there were some challenges to overcome as well. The accumulation of traits that facilitated survival on land may have opened the way to the colonization by plants. Systematists are currently debating the boundaries of the plant kingdom. Until this debate is resolved, we define plants as embryophytes, or plants with embryos. Okay, here are some of the ancestral algae here. Red algae, chlorophytes like sea lettuce, charophytes more closely related to terrestrial plants, and the embryophytes. Okay, plants having an enclosed embryo. These include everything from the mosses all the way up to the redwood tree. And we're going to describe this in greater detail. Key traits that appear in nearly all plants but are absent in charophytes include alternation of generations. This is the way that plants reproduce, a particular method that plants reproduce using two different forms, the gametophyte and the sporophyte. Multicellular dependent embryos. Okay, so spores and embryos are dependent on the parent organism until they are able to attain their own structure. Walled spores produced in sporangia, and apical meristems. These are a form of growing structure, which I'm not sure if we describe in this chapter or maybe in one of the future chapters, plant structure and growth. The gametophyte is a haploid and produces a haploid gametes by mitosis. Fusion of the gametes gives rise to the diploid sporophyte, which produces haploid spores by meiosis. This is known as the alternation of generations, and it's a characteristic of all embryophytes, all terrestrial land plants. Okay, so here we have a basic schematic of the alternation of generations. gametophyte here up at the top. We'll start here. The gametophyte produces haploid gametes by mitosis. It has both male and female characteristics that produce spores. These spores are released into the environment. The egg and the sperm are released into the environment, and then a spore, a gamete from another plant can enter this cycle. This is if this is an egg, then this will be a sperm from another plant, and that sperm will fertilize the egg. That fertilization will give rise to a diploid zygote. That zygote will, through mitosis, begin to develop the structure of a sporophyte. The sporophyte here is the fern structure. The fern structure exists and develops and produces spores through meiosis, and then those spores are released, and these haploid spores. One of those haploid spores may land, and in fertile conditions, it may produce a gametophyte. Okay, the life cycles of all plants alternate between two generations of distinct multicellular organisms, gametophytes and sporophytes. As shown in this diagram, using a fern as example, each generation gives rise to the other, a process that is called alternation of generations. This type of reproductive cycle evolved in various groups of algae and does not occur in the charophytes, the algae most closely related to plants. Take care not to confuse the alternation of generation in plants with the haploid and diploid stages of the life cycles of other sexually producing organisms. Okay, the multicellular haploid gametophyte, the gamete-producing plant, is named for the production by the mitosis of haploid gametes, eggs and sperm, that fuse during fertilization, forming the diploid zygotes. Mitotic division of the zygote produces a multicellular diploid sporophyte, the spore-producing plant. Okay, so meiosis in mature sporophyte produces haploid spores, reproductive cells that can develop into a new haploid organism without fusing into another cell. So this organism here, the gametophyte, is a haploid structure. It produces, through mitosis, it produces gametes and fertilization on through this cycle, through the sporophyte, and again. All right, so this is the alternation of generations. It includes the gametophyte and the sporophyte. Now, down here at the bottom, it shows multicellular dependent embryos. As part of a life cycle with alternation of generations, multicellular plant embryos develop from zygotes that are retained within the tissues of the female parent. The parental tissues protect the developing embryo from harsh environmental conditions and provide nutrients such as sugars and amino acids. The embryo has a specialized placental transfer shell, shown here as a blue outline, that enhance the transfer of nutrients to the embryo through the elaborate ingrowths in the cell wall surface. The multicellular dependent embryo of the land plants, such as this, in such a significant development derived trait that land plants are also known as embryophytes. Okay, so this is the spore-producing cell in a liverwort, and it just shows it just shows greater development of greater independence of what ultimately becomes the spore in the more advanced plants. Okay, here's the alternation of generations. This is on page 506 in your textbook. Multicellular dependent embryos. The multicellular diploid embryo is retained within the tissue of the female gametophyte. Nutrients are transferred from parent to embryo through placental transfers. The land plants are known as embryophytes because of the dependency of the embryo on the parent. Walled spores produced in sporangia. Sporangia are multicellular organs that produce spores. Spore walls contain sporopollen. Remember from the charophyte, it developed the same protein, which makes them resistant to harsh environments. So the land plants are becoming more and more adapted to be able to colonize dry terrestrial surfaces. Okay, each of the many spores produced by a sporangium is encased by a durable sporopollen-enriched cell. Okay, here is a moss, a sphagnum moss. Oh, let's get back here. Okay, here's the sphagnum moss, and hopefully I'll be able to bring some local mosses into the lab, and we'll be able to look at this more closely in class. But here we see the gametophyte, which is the most dominant part of the moss plant life cycle. And then here is the sporophyte, which is the tiny little structure on top of the gametophyte, which includes the sporangium, this little ball at the top. Inside the sporangium is where the spores are produced. So the sporangium is the minor function and the gametophyte is the major function. The gametophyte is dominant and the sporangium is the sporophyte is subordinate, structurally speaking. But the sporophyte produces the spores. Inside the sporangium, this is where the spores develop, and eventually this sporangium cracks open and the spores are released into the environment. Apical meristems. These are parts of the plant that are localized regions of cell division at the tips of roots and shoots. That they're known as apical meristems. If you take a look at any branch, at the tip of the branch, that the end of that branch where the leaf is growing out of, there might be a little bud. Where that bud is the apical meristem on that branch of that bush or tree. You can't see it on the roots, but the roots have the same thing. They have apical meristems, regions that divide through mitosis, and they extend the roots through the soil. Apical meristem cells can divide throughout the plant's life. Additional derived traits include a cuticle, which is a waxy covering on the epidermis that functions to prevent water loss and microbial attack. You can any leaf that you pick up is going to have this waxy covering, this cuticle on it. It's really very apparent. It's particularly noticeable in cacti and other plants that live in the desert because they have to really protect themselves against being dried out. There's a bush called wax myrtle, which is very common in this area, particularly in the coastal plain near the ocean. And wax myrtle has these little black berries on it, and those black berries are coated with a waxy coating. The leaves of this plant are relatively thick, and they have a noticeable cuticle on them. Now, stomata are another derived trait. They are specialized cells that allow exchange of carbon dioxide and oxygen between the outside air and the plant. So these are like little doors that are found all over, particularly the structure of the leaf, and when it's time to draw water into the plant, they open up and they allow evaporation to occur, and this draws water through cohesion, adhesion, tension. You should remember that from Biology 101. Water comes up through the roots into the plant by virtue of the plant opening its stomata and permitting the water to evaporate. Fossil evidence indicates that plants were on land at least 470 million years ago. So just about 500 million years ago, plants begin to colonize the land. Fossilized spores and tissues have been extracted from 450 million year old rock. Here are fossil spores from about 450 million years ago, and this is a fossilized sporophyte tissue. The spores were embedded in the tissue and appears to be from plants. So these bear structural similarities to some modern day plants, so they're kind of like ancient, it's like finding ancient pollen, these spores are. Large plant structures such as the sporangium of Cooksonia. Cooksonia is an ancient plant. It's one of the first vascular plants. It never grew really tall. It was probably just as about the same size as like most of the ferns are today. Ferns are related to bryophytes in some ways. They're related to the mosses and the club mosses in some ways, but they're more related to the Gymnosperms and Angiosperms. They're closer to the more advanced adaptations because they have these they have vascular systems. So Cooksonia did have a primitive vascular system, and it was one of the first plants to colonize the interior regions of the earth. By 400 million years ago, a diverse assemblage of plants lived on land. Unique traits of these early plants included specialized tissue for water transport, stomata, and branched sporophytes. So all of these are traits of that are attributed to terrestrial plants. They're evolving and they're adapting to their new situations with these adaptations. Here's Cooksonia sporangium. This is the sporangium of that plant I was talking about, Cooksonia. I think we're going to see an example of Cooksonia in a little bit here. Let me keep clicking through. Okay, here's Cooksonia. These are the sporangium up on top of it, and they're fossilized in such great condition that you can even see the stomata on them. This is a reconstruction from a 405 million year old fossil. Exhibits dichotomous or the Y-shaped branching with a sporangia at the ends of the branches. This species, Aglaophyton, has rhizoids that anchored it to the ground. They're not true roots. They're rhizoids like the same way that ferns have rhizoids. They anchor them to the ground, but they don't really use them to assimilate nutrients. The inset shows the fossilized stoma of Aglaophyton major. So this very ancient plant. Fungi played an essential role in the colonization of land. Okay, fungi may have colonized land before plants, and they may have adapted to those adaptations, like on the shoreline of some ancient pond, well before plants did. Mycorrhizae are symbiotic associations between fungi and plants that may have developed plants without roots to obtain nutrients. Okay, so plants without roots like Cooksonia and some of the early ferns or the bryozoans like mosses and liverworts, they weren't able to assimilate nutrients without these fungal relationships. And even today, fungal relationships are critical for plants. Basically, every plant has a fungal relationship. It helps them to assimilate nitrogen and other nutrients, and it's very important for the health of the plant. Fungi are heterotrophs, meaning they do not produce their own energy like or use energy from the sun the way autotrophs like plants are. They're heterotrophs, and they absorb nutrients from outside their body. Fungi use enzymes to break down large variety of complex molecules into smaller organic compounds. Fungi can digest compounds from a wide range of sources, living or dead. Fungal walls contain chitin, a strong but flexible nitrogen-containing polysaccharide. The most common body structures are multicellular filaments and single cells like yeast. Well, everybody's probably familiar with yeast. That's what we use to make bread. It helps bread dough rise by the metabolic activity of the yeast imparts carbon dioxide to the bread, and it's also yeast is what helps to produce alcohol in wine and beer. So some species grow as either filaments or yeasts, and others grow as both. Morphology of multicellular fungi enhances their ability to absorb nutrients. Fungi consist of mycelia, which are networks of branched hyphae, which hyphae are kind of like roots. They travel throughout the soil, and a bunch of hyphae together is known as a mycelium. Filaments adapted for absorption. Okay, so hyphae are the filaments adapted for absorption. A mycelium's structure maximizes its surface to volume ratio. So its basic idea is to interface with the soil, the substrate, as efficiently as possible. Okay, here are the mycelium shown in this structure here. Mycelium grow throughout the soil, and the hyphae are the structures, the mats that go throughout the organism, the fruiting body of the mushroom. And this is what. Okay, so what I want you guys to understand here is that the mycelium is the organism. The mushroom is only the fruiting body. So majority of the majority of the organism itself is the mycelium, which grows in the substrate. Now, the mushroom is only the reproductive organ that you see up on the surface of the substrate, and this is what produces the spores and helps to disperse the spores. So these mushrooms will produce spores and release the spores beneath the gills of the mushroom, and the mushroom spores will travel through, with light breezes on the ground, and they will find their way to another substrate that's hopefully advantageous for the organism to grow into a new mat of hyphae into a new organism. So here's some substrate here. These look like pine needles and twigs, and it's covered by the mycelium and the mat of mycelium all together. These are the individual mycelia. The mat altogether is known as the hyphae. Specialized hyphae in mycorrhizal fungi. Okay, mycorrhizal fungi are fungi that have a relationship with plants. Some fungi have a specialized hyphae called a haustoria, which allows them to extract or exchange nutrients with plant hosts. Okay, here is a plant cell on the bottom here, and here this is like a plant rootlet or a root hair, and this is a fungal hyphae that is traveling through the soil. These little structures grow into the cells here, and they begin to communicate with the cells. They're covered with the plant cell will grow a membrane around them, but the hyphae is still able to transmit nutrients across that plasma cell membrane and back into the hyphae, where it then goes into the larger structure, the mycelium of the mushroom. And these structures are called the halcetorium. Halcetoriums grow into these roots. They form little tree-like structures, and those tree-like structures are called arbusculi. I think we'll describe arbuscula a little bit further in the chapter here. So here are the different plant cells, the roots of the plant cell. You can see this is the root growing in this direction, and then it's the fungal hyphae has attached itself to it. Mycorrhizae are mutually beneficial relationships between fungi and plant roots. Ectomycorrhizal fungi form sheaths of hyphae over a root and can grow into the extracellular spaces of the root cortex. Arbuscular mycorrhizal fungi extend hyphae through the cell walls of the root cells and into tubes formed by invagination of the root cell membrane. Fungi propagate themselves by producing vast number of spores. Fungi can produce spores from different types of life cycles. So fungi are actually really diverse sexually. There are not just male and female fungi, but there are like A-type and B-type and C-type fungi. And when different types of fungi come into contact with one another, they can reproduce sexually and produce the haploid mycelium will produce a diploid structure that will live for a short period of time until it produces meiosis of its own and begins to go back into a haploid life cycle. So fungi propagate themselves by producing vast number of spores, either sexually or asexually. Fungi can produce spores from different types of life cycles. Okay, here's the typical asexual reproduction. The mycelium produces spore-producing structures. This is like a mushroom or a tree ear or club fungus or what have you. It's the fruiting structure, the spore-producing structure of the fungus. It produces these spores. These spores germinate. This is all haploid asexual life cycle. Then we have the more complicated sexual life cycle. And in this life cycle, basically you have these haploid mycelium that come into contact with each other. Here's one coming into contact with the other, and they have to be of different types. If they're of the same types, they won't interbreed. But if they are of different types, like type A and type C, they will join together in the process called plasmogamy. Plasmogamy will produce a heterokaryotic stage, which is kind of like a diploid, or it's where these two different types join together. And then there is karyogamy, which phase during which a diploid spore is produced. This diploid spore is produced, and then the diploid spore undergoes meiosis and produces spores with both these traits, the A trait, A type, and the C type traits, and then it produces these spores, and these spores go on, these haploid spores go on through germination. So the diploid stage is just like really short period of time, and the heterokaryotic stage It's not really haploid and it's not really diploid, but it's where the two nuclei of the different types exist in the same form, in the same mycelium, and then they could exist in that stage for days or weeks or even years. Some say even centuries may pass between plasmogamy and the next stage of the sexual cycle, karyogamy. And during karyogamy, the haploid nuclei contributed by the two parents fuse, producing diploid cells. Okay, the zygotes and other structures formed during karyogamy are the only diploid stage in the most fungi. Meiosis then restores the haploid condition. Many fungi then reproduce asexually by growing as filamentous fungi that produce haploid spores by mitosis. Such species are invariably referred to as molds as they form visible mycelia. Other species reproduce asexually and as single-celled yeasts that divide to produce genetically identical daughter cells. Okay, this is complicated, and what I want you guys to know is that these two different types come together, and they join at plasmogamy. They have the heterokaryotic stage. The sexual union occurs in karyogamy. That produces a genetically diverse zygote. That zygote gives rise to meiosis, which produces the spores, and into germination back into the asexual reproduction cycle. It's pretty rare for fungi to do this, but it does happen, and it's an important part of the genetic diversity of the fungi populations. Plasmogamy is the union of cytoplasm from two haploid parent mycelium. Hours, days, or even centuries may pass before the occurrence of karyogamy, nuclear fission. During karyogamy, the haploid nuclei fuse, producing diploid cells. The diploid phase is a short-lived and undergoes meiosis, producing haploid spores. In addition to sexual reproduction, many fungi can and do more frequently reproduce asexually. Molds produce haploid spores by mitosis and form visible mycelia. Single-celled yeast produce asexually through cell division. The origin of fungi. Fungi and animals are more closely related to each other than they are to plants and other eukaryotes. DNA evidence suggests that fungi are most closely related to unicellular protists called nuclearids. Animals are most closely related to unicellular choanoflagellates. This suggests that multicellularity arose separately in animals and fungi. The oldest undisputed fossils of fungi are about 460 million years old. So they arose from separate, distinct protist lineages, but they were closely related. Here's a fossil fungus, 460 million years old during the Ordovician period. Fungi were among the earliest colonizers of land and probably formed mutualistic relationships with early land plants. For example, the 405 million year old fossils of Angiophyton contain evidence of fossil hyphae penetrating cells. This is a plant that looked kind of like Cooksonia. It is, and you can see in these fossil structures here, in these fossil root cells, they show evidence of arbuscular mycorrhizae. Okay, now we'll have a little bit about fungal diversity. Molecular evidence suggests that genes required for the establishment of mycorrhizal symbiosis were present in common ancestor to land plants. Molecular analyses have helped clarify evolutionary relationships among fungal groups, although areas of uncertainty remain. There are about 100,000 known species of fungi. but there are an estimated to be as many as 1.5 million species. The phylogeny of fungi is currently the subject of much research. Most mycologists recognize the 5 major groups are probably paraphyletic. Okay, chytrids are fungi which are they're like yeasts. They form single-celled organisms. They're commonly found in lakes and soil. And the chytrid fungus is there's a process of extinction that's ongoing at the present day, wherein frogs from around the world, amphibians, are becoming weakened and debilitated, and their populations are decreasing and to the point of near extinction. And it's believed that these chytrid fungus are responsible for that because they're freshwater fungus and they can get in under the skin of the amphibians and compromise their immune system. Zygomycetes include certain molds that occur on fruits and breads. This fungi may act as a decomposer if the food is not alive, or parasites or as neutral symbionts. Okay, so these molds occur in a variety of different strategies. They can decompose things, or they can be parasites. Glomerulocytes are the form of arbuscular mycorrhizae with plant roots, supplying minerals and other nutrients to the roots. About 80% of all plant species have such mutualistic partnerships with glomerulocytes. This scanning electron micrograph shows the branched hyphae and arbuscular of the glomerulosmosae bulging into a plant root cell. The plant has been treated to remove the cytoplasm. Okay, so here you can see this is just the cell walls of the plants that remain, and then they chemically removed the cytoplasm, all the organelles and whatnot, and all that remains is the arbuscula of the fungi and the cell wall structure of the plant. Ascomycetes, these are the sac fungi or cup fungi. These are members of a diverse group, are common to marine, freshwater, and terrestrial habitats. The cup-shaped ascocarp fruiting body is of the Ascomycetes shown here gives this species its common name, the orange peel fungus. And Basidiomycetes, these are the most common, the fungus that are most readily apparent and that we're most familiar with. They're often important as decomposers and ectomycorrhizal fungi. Basidiomycetes or club fungi are unusual in having a long-lived heterokaryotic stage in which each cell has 2 nuclei, one from each parent, and the fruiting bodies, commonly called mushrooms of this fly agaric, Amanita muscaria, are a familiar sight in the coniferous forests of northern hemisphere. Now this fungus here, Amanita muscaria, is there are 2 fungus that look almost exactly like it. One of them is used by shamanic peoples of the Siberian area in Russia, and they use it as a hallucinogen to communicate with the ancestral spirit world. But the one that occurs here in North America is deadly poisonous, so do not consume this at any rate. It's a dangerous fungus, but it does have an interesting related species in the old world. Chytrids are found in freshwater and terrestrial habitats. Chytrid have flagellated spores and are thought to have diverged early in fungal evolution. Here's a chytrid fungus here. I'm going to go through these kind of quick because we just went through all that information. Zygomycetes include fast-growing molds and parasites, commensal with symbionts. Here's a Zygomycetes. You can see the sporangia on the tip of the spore there, on the tip of the hyphae. Glomeromycetes form arbuscular mycorrhizae with plant roots. These are the important fungus that helps plants to metabolize nutrients. About 80% of all plant species have mutualistic relationships with Glomeromycetes. Here is the Glomeromycetes in the cell structure. You can see how it got the name arbuscular, because arbor means tree. So an arborist is someone who cuts trees, and a Arbor Day is the day we celebrate tree planting. So an arbuscula is a organ of the fungus that looks like a tree. Ascomycetes live in marine, freshwater, and terrestrial habitats. Ascomycetes produce fungal bodies, fruiting bodies called ascocarps. These are the ascocarps of the orange peel fungus. Basidiomycetes are important decomposers and ectomycorrhizal fungi. The fruiting bodies of Basidiomycetes are commonly called mushrooms. It's the fungus that we're all most familiar with. Here is the Amanita muscaria. Early land plants radiated into a diverse set of lineages. Ancestral species. Okay, so now we're going to go back into plants. Ancestral species gave rise to a vast variety of modern plants. Okay, here is the overall summary of terrestrial plants. They're all coming from an ancestral green algae, a freshwater algae that's related to the charophytes. These gave rise to the liverworts. These are all the non-vascular plants here, the bryophytes, liverworts, mosses, and hornworts. These are all plants with various structural capacity. Mosses have more structure than liverworts. Liverworts just kind of grow out over the surface of a rock or what have you, and they're pretty simple organisms. Mosses are a little more complicated, and hornworts even more so. But all these plants are non-vascular plants. They do not have a vascular system. They do not have a xylem and a phloem, structures within the plant that help move the water and the food, respectively. And as such, they can only attain a minor height. They don't grow more than a few inches tall. Okay, so here's the original common ancestor of land plants. And we go through the mosses and the hornworts, and then we're into the origin of vascular plants. Now, these vascular plants include lycophytes. They do have a vascular system, but it's not very well adapted, so you don't really get gigantic lycophytes, except in the distant past, like during the Carboniferous period, the period when the coal depositions were laid down across the earth. During that time, you actually had lycophytes that were the size of trees. But it was also true that during that time, there was more carbon dioxide in the atmosphere, and it was conducive to the growth of these large tree structures. Since then, they've been outcompeted by other plant species like the angiosperms. Okay, so the manilophytes comes after. It's more advanced than the lycophytes. This includes the ferns. Hopefully we'll get to look at a fern structure in class. Then there are the seed plants, and the seed plants include the Gymnosperms and the Angiosperms. These are all plants that produce an embryophyte that is attached to the sporophyte is the major part of the structure, and the gametophyte is the minor part of the structure, and the gametophyte is the pollen and the egg that join in the cone or in the flower. Okay, here's the origin of land plants a little more closely. This is on page 513 in your textbook. Land plants can be informally grouped based on the presence or absence of vascular tissue. Most plants have vascular tissue for the transport of water and nutrients. These constitute the vascular plants. Non-vascular plants are commonly called bryophytes. That's the liverworts, mosses, and hornworts. Bryophytes are a collection of early diverging plant lineages. Bryophytes are represented today by 3 clades of small herbaceous, non-woody plants. They include the liverworts, mosses, and hornworts. 3 of these three clades are thought to be the earliest lineages diverged from the common ancestor of land plants. Non-vascular plants, bryophytes, okay, that's one lineage there. And then there are the seedless vascular plants, the Gymnosperms, and the Angiosperms. Okay, here are the bryophytes. This is a little closer image of a liverwort. I don't know if you've ever seen these. You find them like around creeks and moist areas. They don't grow very tall. They're just very tiny little plants, maybe half an inch tall. Mosses. We're going to hopefully look at some mosses in class. Here's the gametophyte of the moss, and here's the sporophyte. This whole structure here. And then there's the capsule and the setae. The setae is the stalk on which the capsule sits. And then here is the hornwort with the gametophyte and the sporophyte. Bryophytes are anchored to the substrate by rhizoids, which are not roots. The flagellated sperm produced by bryophytes must swim through a film of water to reach and fertilize the egg. In bryophytes, the gametophytes are larger and longer living than the sporophytes. The height of the gametophytes is constrained by the lack of vascular tissue. Seedless vascular plants, the first plants to grow tall. Bryophytes were the prevalent vegetation during the first 100 million years of plant evolution. Then the earliest vascular plants date back to 425 to 420 million years ago. Vascular tissue allowed these plants to grow tall. Early vascular plants lacked seeds. Seedless vascular plants can be divided into clades. Lycophytes, which are club mosses and their relatives. Manilophytes, which are ferns and their relatives. Okay, so these are the seedless vascular plants. Seedless vascular plants. Okay, here is a club moss. It's a pretty primitive organism. I think they call this like creeping cedar. There's a Lycopodium lucidum, which is a type of lycophyte which occurs in this area around us, and it's the creeping cedar. You may be familiar with it. It has more complex strobili structures which spores are produced, and then of course, so these do have some vascular structure to them. And then the fern, these are the manilophytes. These are the ferns. Hopefully I'll bring in a fern to class too. Okay. There's the lycophyte. The life cycles with dominant sporophytes. In contrast with bryophytes, sporophytes of seedless vascular plants are the larger generation, as in familiar forms. The gametophytes are tiny plants that grow on or below the soil surface. Flagellated sperm must swim through a film of water to reach the eggs. So the Seedless vascular plants are like the bryophytes in that they have a flagellated sperm that must swim through a film of water to reach the eggs. But they are different in that they have a vascular structure. They have a xylem and a phloem, which helps them to grow a little larger. Okay, so here is a breakdown of some of these different plant groups. When we're looking at, we're going to look at the bryophytes, the meliophytes, and the seeded plants, the angiosperms and the gymnosperms. So in the bryophytes, the gametophyte is dominant and the sporophyte is reduced. So here's the dominant gametophyte, the haploid gametophyte, and then the diploid sporophyte is just this little structure on top. In ferns, the plant itself, the leaves of the fern are more dominant. The gametophyte in the fern structure is subordinate. It's just this little organism. You can barely see them. If you look for them in the woods, they're tiny. They're just about the size of your, half of your pinky fingernail. They're tiny little structures. But those are the gametophytes. But they give rise to sperm and eggs, and when the sperm and eggs unite from a different plant, those diploid sporophytes are created. So those sporophytes are more dominant. And in the seeded plants, the sporophyte is vastly more dominant. You can see this example they're giving is like a conifer, like a Douglas fir or a redwood tree, and that sporophyte is the dominant generation right there. The microscopic male gametophytes inside the pollen cone, that is the haploid sporophyte. Okay, and then the microscopic female gametophyte inside the ovulate cone, that is the haploid sporophyte there. So basically, even when you're looking at the cone itself or the strobile, these are parts of the sporophyte. It's only the microscopic egg and sperm that are inside the pollen that are the sporophytes. In angiosperms, similar deal. The sporophyte is much larger. Okay, this could be like an oak tree or a maple tree, and any of the trees that have flowers on them, maybe a tulip popper, or just this little imaginary flower here. The microscopic male gametes are inside these parts of the flower, the stamen, whereas the microscopic female gametophytes are inside the ovules of the flower. So these structures are they're microscopic. The sporophytes are microscopic in the plant, in the seed plants, but they're the gametophytes are microscopic in the seed plants. Okay, so anyways, this table explains it all to you. It's on page 515 in your textbook. Okay, the transport and the xylem and the phloem, this is really what gives the vascular plants their advantage. Vascular plants have two types of vascular tissue, xylem and phloem. Xylem conducts most of the water and minerals that are drawn up through the roots and includes tube-shaped cells called tracheids. Tracheids are technically dead cells. They're laid down by the plant as it's growing. Water-conducting cells are strengthened by lignin. Lignin is the protein that is used to, or is the carbohydrate that is used to strengthen the structure of the xylem. The phloem consists of cells arranged in tubes that distribute sugars, amino acids, and other organic products. Vascular tissue allowed for increased height, which provided an evolutionary advantage. Tall plants were better competitors for sunlight and could disperse spores much farther than shorter plants. Roots are organs that anchor vascular plants. They enable vascular plants to absorb water and nutrients from the soil. So unlike rhizoids are just parts of the bryophytes that they use to make contact with the soil, the substrate, in order to hold them fast into their location. But roots are more evolved than that. Roots are organs that enable them to absorb water and nutrients from the soil. Leaves are organs that increase the surface area of vascular plants, thereby capturing more solar energy that is used for photosynthesis. Leaves are characterized by two types. There are microphylls, which are small leaves with a single vein, like the leaf of a moss, and megaphylls, which are larger, more productive leaves with a highly branched vascular system, like the leaves of a maple tree or an oak tree. Seedless vascular plants were abundant in the Carboniferous period, 359 to 299 million years ago. Early seed plants rose to prominence at the end of the Carboniferous period. Okay, so during the Carboniferous period, conditions were there was a high carbon dioxide and there was a great deal of moisture in the atmosphere, and this was conducive to the seedless vascular plants, like the tree ferns and even the rudimentary Lycopodium, the rudimentary club mosses. Even they even grew to a great height. So some of these tree ferns and Lycopodium, they grew to a fairly decent height. But early seed plants began to rise in prominence at the end of the Carboniferous period, when the land began to dry out and carbon dioxide was less available. Seeds and pollen grains are key adaptations for life on Earth. Seed plants originated about 360 million years ago. An adaptation called the seed allowed them to expand into diverse terrestrial habitats. A seed consists of an embryo and its food supply, surrounded by a protective coat. Mature seeds are dispersed by wind or other means. Living seed plants are divided into two clades: Gymnosperms that have naked seeds that are not enclosed in chambers, and Angiosperms that have seeds that develop inside the chambers called ovaries. Okay, now we're on to the seeded plants, the seeded vascular plants, the Gymnosperms and Angiosperms. In addition to seeds, the following are common to seed plants: reduced gametophytes, ovules, and pollen. The gametophytes of seed plants are microscopic. Okay, you can barely able to view them with the naked eye. The gametophytes are microscopic, which is much of a contradiction to those of the bryophytes, wherein the gametophyte is the dominant stage of the plant. Gametophytes develop within the walls of spores that are retained within tissues of the parent sporophyte. The parent sporophyte protects and provides nutrients to the developing gametophyte. An ovule consists of an egg-producing female gametophyte surrounded by a protective layer of sporophyte called the integument. Female gametophytes develop from a large megaspores. Okay, so here's an example in a ovule from an ovule to a seed in a gymnosperm. In this longitudinal section, throughout the ovule of a pine, which is a gymnosperm, a fleshy megasporangium is surrounded by a protective layer of tissue called an integument. The micropyle, the only opening through the integument, allows the entry of the pollen. So here's where the pollen comes in. Otherwise, the gametophyte, microgametophyte, is surrounded by the integument, protected here. A megaspore develops into a female gametophyte, which produces an egg. The pollen grain, which had entered through the micropyle, contains a male gametophyte. The male gametophyte develops a pollen tube that discharges sperm, thereby fertilizing the egg. Okay, here's the fertilized egg, which includes the embryo. Fertilization initiates the transformation of the ovule into a seed, which consists of sporophyte embryo, a food supply, and a protective seed coat derived from the integument. The megasporangium dries out and collapses. Okay, now the seed is ready for release. The seed can be released into the environment, and because of its condition, it can survive for days or weeks or possibly even years until it finds purchase on fertile soil with the appropriate conditions. Male gametophytes develop from small microspores. Microspores develop into pollen grains, which consist of a male gametophyte enclosed within a protective pollen wall. Pollination is the structure of pollen to the part of the seed plant containing ovules. Pollen eliminates the need for a film of water and can be dispersed great distances by air or animals. A seed develops from the whole ovule. A seed is a sporophyte embryo along with its food packaged in a protective coat. Seeds provide some evolutionary advantages over spores. They may remain dormant from days to years until conditions are favorable for germination. Seeds have a supply of stored food. Okay, so the seed has part of that embryo is used to supply food for the seed. So when the seed germinates, it doesn't have to photosynthesize right away. It can grow for several days or even weeks on the stored food of the endosperm of the cell. If you ever eat coconut, the meaty part of the coconut shell is the endosperm of the coconut. Early seed plants and the rise of gymnosperms. Fossil evidence suggests that by the late Devonian period, some plants have begun to acquire features found in seed plants but did not bear seeds. Gymnosperms appeared in the fossil record about 305 million years ago. Gymnosperms largely replaced non-vascular plants as the climate became drier toward the end of the Carboniferous period. Gymnosperms were better suited than non-vascular plants to drier conditions due to adaptations, including seeds, pollen, thick cuticles on the leaves, and leaves with a smaller surface area. Gymnosperms are an important part of the Earth's flora. For example, vast regions of the northern latitudes are covered by forests of cone-bearing gymnosperms called conifers. This is the region known as the taiga. It is the largest biome in the world, and we're going to talk a little bit more about it when we get into later into the semester. We're going to talk about biomes in global ecology. Okay, so it's the largest biome. It's dominated by conifers. Okay, here's the sago palm. This is a tropical plant right here. It's a gymnosperm. It's not really a palm tree. It's a cycad, which is its own lineage. Here's a Douglas fir. These are large pine trees that are found particularly in the Pacific Northwest. And here's the creeping juniper. These are pretty common as like yard plants around our area, so you might find them. The berries of this low-growing conifer are actually ovule-producing cones instead of fleshy sporophylls. So these are cones instead of fruits here. Down here. Okay, there's the sago palm. typical of tropical areas. The Douglas fir and the creeping juniper. Juniper berries are what make the basis of gin. I don't know if any of you are drinkers, but if you drink gin, that's made from juniper berries. Angiosperms are seed plants with reproductive structures called flowers and fruits. They are most widespread and diverse of all plants. And this is the origin and diversification of angiosperms. The flower is an angiosperm structure specialized for sexual reproduction. Many species are pollinated by insects or animals, while some are even wind pollinated, like corn or wheat. A flower is a specialized shoot with up to 4 types of modified leaves called floral organs. There are sepals which enclose the flower. Those are at the base of the flower. Petals, which are usually brightly colored and attract pollinators. Stamens, which produce pollen. And carpels, which produce the ovules. Now here we're going to see, hopefully in lab, we're going to look at these plant flower structures in a little more detail. You can see here, I'm going to start with the stigma, which is part of the carpel. The carpel is this whole structure here. This is the female part of the flower. The stigma is where the pollen lands, and the pollen grows a little tube called the pollen tube, and it goes down into the ovule of the plant, and it will reach one of these ovules and fertilize it with the pollen grain, and then this is where the gametes of the gametophyte of the female part of the flower, the ovule, the ovary, will become fertilized by the gametophyte of the male flower, which is the cell inside the pollen grain. So the pollen lands here, goes down to the ovule, down through the style into the ovary. The pollen comes from the stamen, which includes the filament and the anther. The filament is always easy to remember because a filament is like a string, or like a filament like in a light bulb. I don't know if you guys are old enough to remember, but light bulbs used to have a little wire inside of them, and that wire was called the filament of the light bulb. So a filament is just like a string or a thread that has some durability to it. So it's, got some it's got some ability to stand up on its own. That's the filament. And then the anther is on the end. The anther is where the pollen is produced. Now, normally what happens this is called a complete flower. It has both male and female parts. In a complete flower, the stigma will be separated from the anther structurally. So hopefully the pollen from here will not pollinate the stigma of this flower. So that will, so they won't have reduced genetic diversity. If they pollinate themselves, which plants can do, then that just reduces genetic diversity. What you want to do is increase genetic diversity. That's the goal of sexual reproduction. So you have pollen produced by another flower or another bush, hopefully, produces pollen, and that pollen gets on the abdomen of a bee, and that bee comes around and visits the stigma of another flower, and then it rubs its abdomen on that stigma and transfers pollen there, and then that pollen from a whole nother bush gets onto this, down through the pollen tube, down into the ovary, into the ovules, and fertilizes the ovules there. And then that's what becomes the seed, is down here. So if you cut an apple across, and you see it's got a 5-part symmetry, those are the ovules of the apple. They will be produced to the seeds. The stamen consists of a stalk called a filament with a sac called an anther, where the pollen is produced. A carpel consists of an ovary at the base and a style leading up to a stigma where the pollen is received. The ovary contains one or more ovules. Seeds develop from ovules after fertilization. The ovary wall thickens and matures to form a fruit. The fruits protect seeds and aid in their dispersal. Various fruit adaptations help disperse seeds by wind, water, or animals. Fruits can function as parachutes or propellers for wind dispersal, like in maple trees, or burrs that cling to animal fur or human clothing. If you've been out in the woods at all, you've experienced burrs. Food that is carried in the digestive system of animals with plants passing unharmed when the animal defecates. In fact, mangoes, if you've ever eaten mangoes, mangoes are delicious. It's one of my favorite fruits in the world. It's a great plant product. Mangoes can only germinate after they've passed through the digestive tract of an animal, typically a cow. So usually, anyway, that's the life cycle of the mango, which is something to think. It's not to detract from the value of the mango itself, but it's just when you think about how these living systems work, it's pretty interesting. Wild cherries, for example, have to pass through the stomach of a bird in order to be in order to germinate. So there's lots of examples of food that's carried through the digestive system of animals, and that because as the animal moves around, it helps that seed to disperse, and then hopefully it'll find its way to favorable conditions, and then it'll be able to produce a new plant. Darwin called the origin of angiosperms an abominable mystery. Angiosperms are the most recent plant lineage, plant clade to occur. They're only about 80 million years old. I think we'll get into this. I may be wrong about that. But they're relatively recent, but they're also the most diverse plant forms in the world. So they're newcomers, but they radiated out quickly, and they took advantage of every adaptation available, and they are more diverse than any other clade of plant. Fossil evidence suggests that phylogenetic analysis have led to progress in solving the mystery, but we still do not fully understand the evolution of angiosperms. Fossil evidence. Angiosperms originated at least 140 million years ago and dominated the landscape by the end of the Cretaceous period 65 million years ago. Chinese fossils of 125-million-year-old angiosperms help us to infer traits that the angiosperm is of a common ancestor. Archaeophractus sinensis, for example, was herbaceous and may have been aquatic. Okay, because some of the most ancient lineages of angiosperms are like the water lilies. Archaeophractus sinensis, 125-million-year-old fossil. This species may represent the sister group to all other angiosperms, or it may belong to the water lily group. Researchers are testing the two hypotheses with phylogenetic analysis. So it's one of those ongoing questions. Angiosperm phylogeny. The ancestors of angiosperms and gymnosperms diverged about 350 million years ago. Angiosperms may be closely related to the benetales, an extinct seed plant with flower-like structures. Okay, here's the benetales. This is a reconstruction showing a longitudinal section through the flower-like structures found in the order Benetales, an extinct group of seed plants hypothesized to be more closely related to extant angiosperms than to living gymnosperms. So this species probably is probably a common ancestor to the angiosperms, having shared derived characteristics that are distinct from the gymnosperms. Ambarella, which is a species, there's only one found now in the South Pacific, but it's an ancient lineage. And water lilies are likely to have descended from the two most ancient angiosperm lineages. So these are the most ancient angiosperms, the Ambarella and the water lily. Okay, Ambarella is a small shrub found only in the South Pacific island of New Caledonia. It may be the sole survivor of the branch at the base of the angiosperm tree. Ambarella lacks vessels, efficient water conducting cells found in angiosperms in later diverging lineages. Okay, so this is like a non-vascular angiosperm, which is a really bizarre thing, which suggests to us that it doesn't have the proper vascular structure, so it's obviously very primitive. Water lilies, species of water lilies in the genus Nymphaea are found in aquatic habitats throughout the world. Water lilies are living members of clades that may be predated only by Ambarella lineage. So the water lily is the second oldest lineage. Star anise, you can find this around here. People grow it in their gardens. Some of these shrubs and small trees in the genus Elysium are native to Southeast Asia, others into the southeastern United States. Living species in the genus probably descended from ancestors whose populations are separated by continental drift. So remember when we talk about continental drift and tectonic plates in the beginning part of the semester, and there are regions where you can find flora and fauna that are similar, that are separated by great geographic distances. Southeastern United States is very similar to southeastern China. There are alligators in the southeastern United States and alligators in China. I mean alligators that are distinct from crocodiles. Crocodiles and alligators are different, and alligators are found basically only in southeastern United States and southeastern China. The star anise also has the same distribution. There is a plant called ginseng that has a similar distribution found in China and in the United States. So we can infer from that a long period of time ago there was biological connectivity. These continents were probably much closer together, perhaps even touching, in order for these species to become dispersed onto those different locations. Okay. The next oldest lineage is the magnoliids. Everybody knows the magnolia trees here around in the southeast. We've got magnolia trees. It's one of the greatest things about springtime in this area. It produces these large, fragrant flowers. They're some of the oldest trees, the oldest lineage of trees around. This clade consists of about 8,000 woody and herbaceous species, including the familiar and economically important magnolias, laurels, avocado, cinnamon, and black pepper. A variety of southern magnolia here, Magnolia grandiflora, has flowers that can measure up to about a foot across. So they have gigantic flowers. And monocots, all right, these are, lilies and irises and a lot of your bulb plants, tulips that grow in the garden. They have straight venation in their leaves. They have a linear parallel venation in their leaves. That's distinct from the eudicots, which have net venation in their leaves. But the monocots are plants like bananas and all kinds of different things, rice, corn, wheat. Other monocots are used as ornamental plants. Monocots include orchids, grasses, irises, onions, garlic. Monocots are pretty diverse, but they pale in comparison to the diversity of the eudicots. Eudicots are nearly 70% of all living flowering plants are eudicots. One example here, the zucchini, is a subspecies of cucurbita. Anyway, it's zucchini, an important crop, as are acorn squash, pumpkin, and other species. Eudicots also include southern sunflowers, cacti, clovers, oaks, and a wide range of other species. Okay, here's the close-up of the table. You can see the eudicots. They're quite diverse. Eudicots are 70% of all species, and they've only evolved 125 million years ago. They really radiated out about 100 million years ago, and they compose the majority of the floral fauna. Ambarella includes only one known species, a small shrub called Ambarella trichopoda, which is found in the South Pacific on the island of New Caledonia. Water lilies are found in aquatic habitats throughout the world. My parents even have water lilies in their backyard. It's common, you can have a garden pond, ornamental pond, and typically water lilies are grown there. And then the second oldest lineage of flowering plants in the world. So it's pretty amazing to think about when you're sitting there by the pond. Star anise naturally occur in Southeast Asia and southeastern United States. Extant species are likely descended from ancestral populations that were separated by continental drift. Magnoliids, including magnolias, laurel, avocado, cinnamon, and black pepper plants. Monocots account for more than 1/4 of angiosperm species. Eudicots account for more than 2/3 of angiosperm species. Land plants and fungi fundamentally change chemical cycling and biotic interactions. The colonization of land by plants and fungi altered the physical environment and organisms that live there. A lichen is a symbiotic association between a photosynthetic microorganism and a fungus. Lichens are important pioneers on new rock and soil surfaces. They break down the surface, affecting the formation of soil and making it possible for plants to grow. Lichens have helped the colonization of land by plants. I'm not sure if they go into the evolutionary history of the lichen here. I think it's probably pretty complicated. Okay, here are different kinds of lichen. There's crustose lichen that grows on the surface of a rock, and foliose lichen also grows on the surface of a rock. One thing that's interesting about lichens is they are the first stage of primary pioneering of a ecosystem. So this is like the first stage of primary succession. So you have nothing but barren rocks in a volcanic field. The next thing you get is lichens. And these lichens start to grow on these rocks. They find little cracks and crevices to grow in. And then through their action of their rhizoids, they begin to break apart the surface of the rocks, just bit by bit, loosening up the mineral surface of the rocks. And then the mineral surface of the rocks begins to break down a little bit, and it forms pockets of rudimentary soil that can be exploited by the mosses. So the mosses are the next thing to come in. And then the mosses come in and they begin to inhabit those areas that the lichen has prepared for it. And then the mosses break up the soil, break up the surface of the rock even more. And then the mosses die on top of themselves and they form organic soil. And that organic soil can be exploited by ferns. So even when we're talking about like the evolutionary history of these organisms, you can see how something as primitive as a lichen could come and exploit the barren surface of the earth and then break it down a little bit by a time and give rise to mosses, and then mosses could give rise to ferns. So over time, we're seeing the same kind of this colonization of the land is like primary succession of the ecological system. Well, I think we're going to talk about primary succession in chapter 43, maybe. I'm not sure where we're going to get into it, but hopefully we'll talk about it a little bit in class if we don't get around to it in the textbook. Okay, here's the anatomy of lichen. It shows the Ascomycetes fungus here, and then it's got little algal cells wrapped up in it, and that's what gives the green color to the fungus here. So this could have been, lichen could have been one of the earliest organisms to colonize the surface of the earth. Lichens are neat. You can find them if you go for a walk in the woods. You can find them all around. They grow on trees. Where they grow on trees, it's an indication of good air quality. If you have trees in your yard that have lichen, it suggests that you have good air quality in your area. Plants affect the formation of soil. Roots hold the soil in place, and leaf litter and other decaying plant parts add to the nutrients of the soil. Plants have also altered Earth's atmosphere by releasing oxygen to the air through photosynthesis. So remember that during the Carboniferous period, there was a lot of carbon dioxide, and it was allowing those club mosses to grow really big into tree-size trees, and we had tree ferns and all kinds of lush vegetation. Well, what one theory is that all that vegetation used up the carbon dioxide, and the process of using up the carbon dioxide, it promoted more oxygen into the atmosphere. That more oxygen is what we breathe today. So it's part of our environmental system, part of our ecology. Plants and fungi affect the cycling of chemicals in ecosystems. Plants absorb nutrients which are passed on to the animals that eat them. Decomposers, including fungi and bacteria, break down dead organisms and return nutrients to the physical environment. Plants play an important role in the carbon recycling. Photosynthesis removes carbon dioxide from the atmosphere. Increased growth and accelerated photosynthesis resulted from the formation of vascular tissue and may have contributed to the global cooling at the end of the Carboniferous period. Okay, look, here's the lycophyte trees that I was talking about. Here's this artist's rendering of the ancient Carboniferous period. Here's ferns, tree ferns, the lycophyte trees, tree trunk covered with small leaves, like these are micropiles on the trunks of these trees. And the lycophyte tree reproductive structures, they might look kind of like sporangia, like Cooksonia or something like that. And here's a little dragonfly. It's suspected that during the period of the Carboniferous era, dragonflies may have attained a size of up to a foot across. So because the conditions of the atmosphere were different, they were able to grow larger. There was greater, towards the end of the Carboniferous period, there was a greater amount of oxygen present. Biotic interactions can benefit both species involved, called mutualisms, or benefit one species while harming the other, as when a parasite feeds on its host. Plants and fungi have large effects on biotic interactions because they increase the available energy and nutrients on land. Mutualistic fungi absorb nutrients from a host of organisms and reciprocate with actions that benefit the host. Plants harbor harmless symbiotic endophytes, fungi that live inside leaves or other plant parts. Endophytes make toxins that deter herbivores and defend against pathogens. Okay, so here in this case, here are some endophytes that are existing inside the plant leaves of Theobroma cacao. This is the tree that gives us coffee. Here is leaf mortality. Endophytes present, not present. So without the endophytes, leaf mortality is increased. Leaf damage is increased. With the endophyte, it's pathogen resistant, and you have less mortality. So here's the fungal relationship. The endophyte is positive here, it's positive here, reduced leaf mortality, reduced leaf area damage. That's on page 523. It's one of those inquiries. Parasitic fungi absorb nutrients from host cells but provide no benefits in return. About 30% of known fungal species are parasites or pathogens, mostly on or in plants. For example, Cryphonectria parasitica causes chestnut blight. There used to be that chestnut trees were all across the eastern part of the United States. They were a vital part of the economy, and people used to let their pigs run wild, and they would feed on acorns and chestnuts. But then this blight came over about 150 years ago or so, and it wiped out all of the chestnut trees. So it is a pathogenic parasite. It destroyed the chestnut trees. Okay, here's corn smut. Corn smut is a fungus that attacks corn. It's actually in Mexico. People, they have festivals and they eat the corn smut. So it is edible. They cook it up with herbs and spices, and apparently it's delicious. I've never had it. Tar spots fungus on maple leaves. You can see this in this area. And these are ergots, which are a type of fungus that grow on rye. And ergots are interesting. They produce the chemical lysergic acid, which is the basic component of lysergic acid diethylamide, which is LSD, a powerful hallucinogen. And it's believed that when they had the witch hunt trials in Massachusetts back in the 1700s, when the Puritans got all excited about being witchcraft and people flying around on brooms and whatnot, it's believed that these ergot, they may have gotten some of this ergot in their rye and used the rye in their bread and then eaten the bread and hallucinated. They may have hallucinated these events that led to the witch trials. Corn smut, delicious, yummy. Tar spot fungus, ergots, lysergic acid. Animals influence the evolution of plants and vice versa. For example, animal herbivory selects for plant defenses. For example, interactions between pollinators and flowering plants select for mutually beneficial adaptations. Obviously, a plant like an orchid flower is usually evolved for one specific species of orchid bee, and it's only that orchid bee that can pollinate that flower. So they have a co-evolve. They co-evolve into those factors. Other flowers have a different strategy where they try to be available to pollination by as many different species as possible. Clades with bilaterally symmetrical flowers have more species than those with radial symmetrical flowers. This is likely because bilateral symmetry affects the movement of pollinators and reduces gene flow in diverging populations. Bilateral symmetry, okay, more specific. Radial symmetry, you can see it's more open to different types. Time since divergence of common ancestor, and compare the number of the different species. Okay. On average, the clade with bilateral symmetric flowers has nearly 2,400 more species than did the closely related clade with radial symmetric. Okay, so a key step is to identify cases in which clade, which bilaterally symmetric flowers shares a immediate common ancestor with a clade of flowers who has a radial symmetric flowers. One recent study identified 19 such pairs of closely bilateral and radial clades, and on average, the clade with bilateral symmetry had nearly 2,400 more species than did the closely related clade with radial symmetry. This suggests that flower shape can affect the rate at which new species form, perhaps because how flower shape affects the behavior of insect pollinators. Okay, this is complicated. Let's not worry about it too much. I think we're going on 2 hours here, so I'm going to try to wrap this up. Angiosperms and other plant groups are being threatened by the exploding human populations and demand for space and resources. About 55,000 kilometers square of tropical rainforests are cleared every year. That's about half the size of the city of Richmond every year, and it's mostly in the Amazon rainforest, although there is some deforestation going on in Africa, but it's a sizable amount. Deforestation leads to the extinction of plant, insect, and other animal species. Okay, so if you eliminate plants, you're extinguishing plants, insects, and other animal species. It's all connected. If current extinction rates continue, more than 50% of Earth's species will be lost within the next few centuries. Here's a satellite of deforestation in Brazil. The top one is the earlier image. The bottom one is the later image. In 10 years, there's this much deforestation that has occurred. Okay, here is different treatments of soil, and this is a metric of how well the plant is growing. So the plant grows best when there is a treatment of the soil with the fungus. So here we have the fungus treatment. Plant is growing well. Plant is not growing well with no fungus treatment. Okay, this is our vascular fungus forming a mycorrhizal relationship with the plant. It's not with the fungus. The plant grows better. Okay, here's a table of that same information in a different format. Okay, this shows that these are roots growing in different soils at different temperatures. The fungus helps the root to be more healthy in a higher temperature. Without the fungal relationship, the root is not healthy. Okay, gametophytes, alternation of generations. You got to know that. You got to know the parts of the flower. Got to know that. Flower anatomy. You want to know the basic structure of the phylogenetic tree of the terrestrial plants. Okay, here's soil temperature and different fungi and fungus relationship. Okay, this shows, the plus shows with the fungus relationship. The minus shows without the fungus relationship. Okay, so the plant basically died when the temperature was high. The plant died when the temperature was low. The plant grew better with the fungus. Okay, so the plant grows well with the fungus. When the temperature is low, it grows better. Okay, so here's without the fungus. It grows 16 grams with the fungus. Okay, anyway, this is the last slide. If you want more information about this, it's an exercise in your scientific inquiry on page 527. This is a long and complicated chapter. Do your best. We're going to go over it in lab. If you have any questions, send me an email or contact me by phone if it's an emergency. If you're having trouble with your getting online, that's an emergency. If you have a question about the information in the chapter, you can send me an email. Okay, I look forward to seeing you again at lab.